Histology, that is the most neglected yet the most scoring topic. The common things heard in a histology practical lab are अरे सब सेम ही तो दिख रहा है, everything is blue pink only and they are different hues. मुझसे नहीं होगा ये सब. And if you are also thinking like this, then you are at the right place. Hi everyone, this is Pratiksha Paul, a second year MBBS student at GMC Sundargarh. And today we are going to deal with the topic that is the histology. In today's video, we are going to discuss about the basics of histology and how you can identify a slide. इनिशियली मुझे भी सब ब्लू पिंक ही दिखता था और मैं भी कहती थी कि मुझसे नहीं होगा, ये सब छोड़ ही देती हूँ. लेकिन slowly I do get the hold of the subject and today I am going to share my experience with you all. There are some unsaid rules of histology. Number 1. Do not directly jump into the systemic chapters. Learn to identify the basic tissues first, like the epithelium, the muscle, the cartilages, the glands, etc. And if you can identify them, then your job is half done. Do not look into the microscope vaguely. Start looking the slide from one end, like from the epithelial side, and then go deep. And you can, if you can identify the epithelium, then you can remember this epithelium is present in this structure, and you can identify the structure also. Number 2, read the theory of the histology slide that you are going to look into the microscope. Every slide has its identification features. Keep them in mind while looking in the microscope and try to focus on them. Then you can easily identify the slide. Number 3, draw the histology diagrams in your journal sincerely and do not draw it for the sake of completion. It will help you eventually in your exams a lot. And diagram should be technically correct and सुंदर नहीं भी होगा तो भी चलेगा. Number 4, click the original microscopy slide pictures in your practical classes. It will help a lot in your exam time. Now that all of you have know the thumb rules of histology, let's get into the basics of histology. That is, today we are going to read about the epithelia, the muscular tissue, and about the cartilages. Before going into the topic proper, please hit that subscribe button. That will make up for all the time and effort I put into these videos. Thank you. So let's get into today's topic. Today we are going to learn about the basics of histology. So the first thing is microscopy, because तुम slides microscope में ही देखते हो. So it is not very much important to learn about the microscope in histology, but in physiology it is asked in the first year. So the microscope through which you are seeing the slides is a light microscope or a compound microscope. It is also known as the bright field microscope. You should know the parts of microscope. So the uppermost part is the ocular lens or the eyepiece through which you are seeing the objects, and below which there is a nose piece, then the objective lens, then there is a stage upon which you are putting your slides, then below the stage there is a condenser and a diaphragm through which you can adjust the light, and below that there is a light source. The back of the microscope is known as arm, that is the support, and there are two adjustments. One is fine adjustment and one is coarse adjustment through which you can adjust your slide. Then the lowermost part is the base. So the next basic thing you have to know about is the staining. Staining procedure is not as such important, but you have to know which stain is used and what are its properties. So the stain used for preparing the histology slide is the hematoxylin and eosin stain or the HNE stain. The hematoxylin stain is a basic stain. It gives a purple color. It stains the nuclei, that is the DNA RNA part. So you can see here beautifully the nucleus are the purple in color. And eosin stain is a acidic dye. It gives a pink color. So it stains the cytoplasm, that is mostly the protein part. So here you can see the cytoplasm is perfectly pink and it's looking beautiful. Here in the this is the epithelial layer, but in the below you can also see the nucleus is purple and the muscle fibers or cytoplasm is pink color. Mucous cells like goblet cells look almost clear because they don't take up the hematoxylin and eosin stain. Here you can see the goblet cells that are present in the intestine are looking almost clear because they don't take up the HNE stain. So if there is a clear space, you can identify that it is a goblet cell, and goblet cell are present in intestine. So you can say that the slide is of intestine. Now coming to the epithelial tissue, you have to know the classification of the epithelial tissue. So the epithelial tissue is classified into two types, that is the simple epithelium and the compound epithelium. Simple epithelium is unilayered and the compound epithelium is multilayered. Simple epithelium is again classified into three types, that is the squamous, cuboidal and columnar epithelium. Columnar epithelium is again classified into two types, that is the simple columnar and ciliated columnar epithelium. Now, compound epithelium is classified into stratified and transitional epithelium. Stratified epithelium is again divided into three types, that is the squamous, cuboidal and the columnar epithelium. And the squamous epithelium is again divided into two types, that is the compound squamous keratinized and squamous non-keratinized epithelium. So this is the classification of the epithelium, and these are all the epitheliums that you will find in different body parts. And by identifying the epithelium, you can understand that it can be such and such structure, because some epitheliums are confined to some structures only. So you can easily identify the slide. Now coming to the individual epithelium, we are going to discuss about all the epitheliums separately. So first coming to the simple squamous epithelium. Simple squamous epithelium is single layer, because all the simple epithelium are unilayered only. So it is a single layer flattened cells. You can see here the single layer flattened cells. These are the squamous epithelial cells, and the height is less than the length, because it is more elongated but not very high. So these are the cells that have the height less than the length. The nuclei produce a bulging in the surface, and you can clearly see here the nuclei produces the bulging towards the surface. This is a endothelial lining. Endothelial lining have the simple squamous epithelium. You can see these are the squamous epithelial cells, and these are the RBCs present in the endothelium. So where can you find simple squamous epithelial cells? These are found in the alveoli of the lung and the endothelial lining. There are other examples also, but you do not have to remember every example. Remember only the important ones. Now coming to the simple cuboidal epithelium. Simple cuboidal epithelium are single layer cube-like cells. I told you simple epithelium means it is unilayered only. So the cuboidal cells are, the name suggesting, they are the cube-like cells. Cube-like cells, and it has the centrally placed nucleus. Centrally placed nucleus, you can see here. These are the cuboidal epithelial cells. The nucleus is placed at the center. If you are wondering, it is a slide of thyroid gland, and the thyroid follicles are lined by cuboidal epithelial cells. So you can see these cuboidal epithelial cells beautifully lined the thyroid follicles. And the function of the simple cuboidal epithelium is secretion, absorption and excretion. So here the thyroid follicles are secreting thyroid hormone, so the lining epithelium is the cuboidal epithelium. Where can you find simple cuboidal epithelium? Thyroid follicle, as I told you, and the ducts, epithelium of ovary and proximal convoluted tubule. The secretion, absorption, excretion function, where it is happening, all those places you can find simple cuboidal epithelium. Let's get into simple columnar epithelium. Simple columnar epithelium is layer of tall cells. Tall cells, you can see these are the simple columnar epithelium. These are tall cells. How much tall is this cell? And this is the nucleus. This is the cytoplasm. The nucleus is presented at the base. In every cell, the nucleus is present at the base, not in the center, not in the periphery. It is present in the base. Now we can easily identify simple columnar cells. So other features are the nucleus is oval, elongated and present at the base. As I told, this epithelium has its function like secretion and absorption, similar to cuboidal epithelium cell, but there is not the excretion property. Simple columnar cells are found in the stomach, large intestine, and ciliated columnar cells are found in the uterus, fallopian tube, and the vas deferens, where it propels the ovum or the sperm. So, columnar epithelium are of two types: simple columnar and simple ciliated columnar. So, simple columnar is found in the stomach and large intestine, and ciliated columnar is found in the uterus, fallopian tube and the vas deferens part. So we are finished with the simple epithelium, that is simple squamous, simple cuboidal and simple columnar epithelium. Now let's compare these three together: simple squamous, simple cuboidal and simple columnar. In squamous, the cells are flattened and the nucleus is more elongated, and in cuboidal the cells are cube-like and the nucleus is centrally placed. You can see this nucleus, these are centrally placed. And in the columnar cell, the cells are tall and the nucleus is placed at the base, and oval and elongated nucleus are present. And you have to remember the examples of where the epithelium is present. As we are done with the simple epithelium, now let's get into the compound epithelium. So in the compound epithelium, the first thing is the stratified squamous epithelium. Stratified squamous epithelium is of two types: stratified squamous keratinized and stratified squamous non-keratinized. So what are the features? It has several layers, multi-layered, and the most superficial cells are flat, and the deepest layer is columnar and it rests on the basement membrane. So here you can see the most superficial layer is flattened, but the deepest layer are resting on the basement membrane. And what are the nuclear features? The nucleus is oval in basal layer, round in the middle layer, and transversely elongated in the superficial layer. Here you can see the nucleus is oval in the basal layer, round in the middle layer, and transversely elongated in the superficial layer. So what is the function of the stratified squamous epithelium? It is protection and prevent dehydration. As it is multilayered, it provides us the protection. So where can you find the stratified squamous epithelium? The non-keratinized one found in the esophagus and vagina, and the keratinized one is found in the epidermis of the skin. So in the keratinized epithelium, the surface layer is made up of keratin. Here you can see the keratin layer is a thick keratin layer present in the stratified squamous keratinized epithelium. Other things are same as the stratified squamous non-keratinized epithelium, but only the surface is covered by the keratin. Next, coming to the transitional epithelium. The transitional epithelium is a multi-layered epithelium with round nucleus, and it is exclusively located in the urinary epithelium. So it is also known as the urothelium. Remember, it is very important. It is asked in the exams also. The transitional epithelium is also known as the urothelium. So here you can see the transitional epithelium. It is multi-layered and the cells are present one on the other and with the round nucleus. Other epitheliums like of the compound epithelium, like the stratified cuboidal and stratified columnar, are same as simple cuboidal and simple columnar. The only thing that is different is the simple epithelium is of unilayer, but the compound epithelium is of multilayered. So where can you find the transitional epithelium? It is present in the renal pelvis, the urethra, urinary bladder, ureter, etc. So if you can identify transitional epithelium in a slide, you will never say that it is a slide of GI, because transitional epithelium is present only in the urinary tract. So the structure has to be a ureter or urinary bladder. You cannot make these blunder mistakes here. That is why identifying the epithelium is important. So now everything about the epithelium is over. Now if you are identifying the stratified squamous epithelium somewhere, then it must be the esophagus if it is not keratinized. You cannot make blunder mistakes if you identify the epithelium easily. Okay, so everything about epithelium is done. Now we are going to deal about the cartilages. So there are three types of cartilages: hyaline cartilage, elastic cartilage and fibrocartilage. This is the table from Ivy Singh book. So these are all the things you need to know about the cartilages. Hyaline cartilage, elastic cartilage and fibrocartilage, these are the differences of their features. You have to know about them because it is asked in the exams. So the appearance of hyaline cartilage is of ground glass type, while fibrocartilage is white. And the main cells of the hyaline cartilage is chondroblast, chondrocyte, and present in the isogynous groups. But in elastic cartilage, cells are present in single or small groups. But in fibrocartilage, the cells are arranged in a row. Like this, the fibers present in the hyaline cartilage are of type 2 collagen fiber. In elastic cartilage, type 2 collagen fiber and elastic fibers are present. But in fibrocartilage, both type 1 and type 2 collagen fibers are present. And about calcification, in hyaline cartilage calcification occurs. In elastic cartilage there is no calcification, but in fibrocartilage calcification occurs only during the repair process after any injury. And the most important thing to remember is their distribution, where you can find these cartilages. So the hyaline cartilage is found in the most importantly the trachea, large and smaller bronchi, costal cartilages, thyroid and cricoid cartilage, and the base of arytenoid cartilage. You do not have to remember every example I am telling you. The important things are here: the trachea and the bronchi, and also the epiphysis or articular cartilage. These are the important examples. Like that, in elastic cartilage you can remember epiglottis. कंफ्यूजन होता है कि ट्रैकिया और एपिग्लोटिस में कि कौन सा इलास्टिक है एंड कौन सा हाइलेन कार्टिलेज है. सो याद रखने के लिए इजी है कि इलास्टिक कार्टिलेज ई से शुरू होता है एंड एपिग्लोटिस ऑल्सो स्टार्ट फ्रॉम ई. सो एपिग्लोटिस इज इलास्टिक कार्टिलेज. And fibrocartilage is present in the intervertebral disc, symphysis pubis, like this. So, starting about the hyaline cartilage, what are the salient features? I have already told. The isogenous group of chondrocyte present as cell nest. It appears translucent or glossy, separated by homogenic basophilic matrix. This is important to identify basophilic matrix. This basophilic matrix is not present in the elastic cartilage. Chondrocytes increase in size from periphery to center. You are in the exam you are given to write two identification features about the slide. If you are given a hyaline cartilage slide, then you can easily write these 3 points. And the important example is it is present in the trachea. So here you can see the hyaline cartilage. These are the chondrocytes that are present in the groups, and you can see the basophilic areas around it. So the cells are surrounded by a basophilic matrix. You can easily identify it as a hyaline cartilage. And if there is a slide of trachea or epiglottis, if hyaline cartilage is present, you can easily identify it as a trachea. Next, coming to the elastic cartilage. So, elastic cartilage is characterized by single chondrocytes present in a lacuna and surrounded by bundles of distinctly visible elastic fiber. Because with collagen fiber in elastic cartilage, elastic fibers are also present. And the principal site is epiglottis. Not the principal site, the main example you have to remember is the epiglottis. So, elastic cartilage is seen something like this. These are the chondrocytes present in the lacuna, and these are the elastic fibers that are surrounding the lacuna. Okay, so hyaline cartilage and elastic cartilage is done. I am not telling you about the fibrocartilage because these two are the most important, because you have slides of trachea and epiglottis in your systemic slides. Now our epithelium and cartilage is done. Now coming to the last one, that is the muscular tissues. Muscular tissue are of 3 types, that is the skeletal muscle, the cardiac muscle, and the smooth muscle cell. So let's learn about the skeletal muscle first. So the skeletal muscles are, as you know, it is a voluntary muscle. So the fibers are long and parallel. No branching is present in the fibers. The nucleus are flat and peripherally placed in the muscle fiber, and it has a transverse striation present all over, that is the A band and I band. So you can see here the skeletal muscle. Beautifully the transverse fibers are seen here. These are the transverse fibers that are present, and the nucleus are present in the periphery. Nucleus are present in the periphery, not in the center, and there is no branching present. So you can easily identify it as the skeletal muscle. Now coming to the also it is a longitudinal section. If you are taking a transverse section, then you can see the fibers like this. These are the fibers, and the nucleus is present in the periphery like this. Okay, so now coming to the cardiac muscle. In the cardiac muscle, the muscle fibers branch. This is the differentiating feature between the skeletal muscle and the cardiac muscle. here the muscle fibers branch and anastomose with each other. And the muscle fibers is a single nuclei that is centrally placed. In skeletal muscle, the nuclei is placed peripherally, but in the cardiac muscle, the nuclei is centrally placed. And the adjacent cells separate from each other by intercalated disc. Intercalated disc is only the feature of cardiac muscle. So here you can see these are the intercalated disc present in the cardiac muscle, and the muscles are branching and anastomosing into each other. So this is the cardiac tissue, and the nucleus is present in the center, not in the periphery. The nucleus is present in the center of the muscle fiber. Okay, so this is the cardiac tissue. Now coming to the smooth muscle. Smooth muscle is the spindle shaped cell, and you know it is the involuntary cell. So spindle shaped and with tapering ends. How it is looked like this. It is a spindle shaped with tapering ends, both side tapering ends. And the nucleus is elongated and centrally placed. Nucleus is also elongated and centrally placed like this. So here you can identify in the slide, these are the spindle shaped cells and the nucleus is present in the center. So here you can differentiate between the three muscular tissue, that is the skeletal, cardiac and the smooth muscle. In skeletal muscle there is striations present, but in cardiac and smooth muscle there is no striations, and in the skeletal muscle there is no branching, but in cardiac muscle branching is present, and the nucleus is present peripherally in skeletal muscle, but in both cardiac and smooth muscle the nucleus is present in the center. So these are all the basic things that you need to know about the histology. Also, I missed the gland part here. I will put it in the next part of this histology chapter. So these are all the basic things that you need to know about histology to identify the slides easily. Once you identify these basic things, तुम्हारे माइंड में ये आएगा कि हाँ ये एपिथेलियम है तो ये चीज हो सकता है ये स्लाइड, तुम ब्लंडर मिस्टेक्स नहीं करोगे. Okay, so that's all from my side for today's video. I will be coming with systemic slides also in the next part of this histology chapter, and I hope these things will help you in identifying the slides in your practicals, and I hope I can make you familiar with the subject. So thank you so much for watching, and please make sure to like, share and subscribe. Thank you.